Okay, welcome back everybody. Hope you had a good break. So welcome back to the second part of day one of the Analysis for Authenticity conference. Hope you're enjoying it so far. I certainly am. So we've got 3 more new speakers for the rest of this afternoon to hear from, and then we'll have a Q&A session at the end. So do keep posting in your questions into the Q&A link, and we'll pick them up at the end. So our next speaker is also from Ferroscience, so it's my pleasure to welcome Rosario Romero. Nice to see you again, Rosario too. Rosario is the science lead for novel foods, and today she's going to be telling us about some of the different analytical tools we have used to support the verification of alternative proteins, which is a very topical and interesting area. So Rosario, over to you. Welcome.
Thank you, Lucy. And thank you, organisers, for inviting me to speak here today. Can I just check that you see my screen properly and you can hear me?
Yes, I can.
Yeah. Okay. Great. Thank you. So yes, I'm going to talk about analytical tools, and this presentation is really a summary of the outputs of a project that we completed 2 years ago. Sorry, I'm trying to move to my next slide. That is not right now. So yes, it's a project that we at Ferroscience did for DEFRA, completed a couple of years ago, and is now published on the DEFRA website. And what we did in the project was, first of all, a high-level overview of current innovations in the alternative protein space, to get a sense of the type of products that companies are developing at the moment, and that might be in the market in the coming years. So once we had that idea, we used those types of products to focus literature review, and we also had interviews with the relevant stakeholders to really explore what the potential food authenticity implications are in relation to alternative proteins, whether there are new risks that we need to take into account, and also consider the current methodology that we have now, and whether or to what extent new current technologies are suitable, or what capabilities we need to develop to make sure that we are prepared when novel products start coming to market. So just a little bit of background. This is only to show that the interest in alternative proteins continue to grow. This is just an example of a graph from a publication, but all projections agree that this will continue to grow. And of course, the main drivers are reasons of food security, with a global population that continues to increase, and the need for more sustainable foods that don't have so much of an impact on the environment. This is really an attractive option. There are also reasons related with health perceptions and animal welfare associated with livestock production. So strong reasons behind, and the trend continues to grow. There are lots of different innovations. These are some of the main categories of novel alternative proteins that people are working on. They include plant, algae, insect, cell cultivated products, fermentation type of production, either biomass fermentation or single cell proteins, or precision fermentation, where we use microorganisms, for example, to produce proteins of interest. Some of these will have the potential to impact the food system, but there are still lots of challenges that the industry is facing. First of all, optimizing technology, and also scaling up. This is a big step that they need to give, because usually the technologies may be working well at laboratory level, but scaling up to the levels that are needed to have an impact on the food system is another matter. Then there are other challenges, like achieving price parity, consumer acceptance in certain categories that may be a little bit more difficult. There are regulatory aspects, sustainability credentials to worry about, and also making sure, of course, that the nutritional profile is adequate and that foods that are being produced are safe. And then there is food authenticity. From the project that we conducted, we realized that there hasn't been a lot of focus on food authenticity in relation to alternative protein development. And this, I suppose, is logical because of all these challenges that industry are focusing on. But certainly regulators, and that's the reason why DEFRA funded this project. It is certainly a good time to start thinking about food authenticity and any emerging risks that might be associated with alternative proteins, and also how we can face those challenges and provide consumer confidence. Food authenticity and food fraud are old issues. This has always existed, and it is more likely to happen when the complexity of the food supply chain increases, and that may be the case with alternative proteins, at least with certain categories. The main driver, as we know, is economic gain, and this can lead to cases of manipulation of the food, so deliberate adulteration of foods by either substitution or addition, dilution, different types of adulteration, but also cases of mislabeling where the product is misdescribed, or there is a failure to declare ingredients or to declare the production methods, or falsification of the country of origin, for example. So lots of different types of fraud. And when we think about alternative proteins, we can envisage situations where the risks might be higher for alternative proteins. For example, if we're thinking about substitution, adulteration, where cheaper ingredients are used instead of the original intended ingredient. Alternative proteins are more expensive to produce, at least at the beginning, until that price parity is achieved. So substitution for conventional counterparts can be happening, or substitution for another lower value protein. Adulteration with powders is something that came up as a potential risk as well, because some of these proteins are produced in powder form to be used as ingredients in products, sometimes hybrid products where it's not easy to recognize the ingredients. So these are potentially more vulnerable to fraud. When the supply chain is complex and then there are disruptions, for example, excess of commodity or shortages of certain ingredients, these may lead to substitution as well. And again, in the case of alternative proteins, with so many novel ingredients, novel processes, and the complexity of the supply chain, there may be potential for disruption. And there's also the issue of competition. This came up as well as a factor that may be putting a lot of pressure in some cases where there are many companies developing competing products, and there's a lot of stake for many of them that have invested heavily on developing technology and getting all the regulatory requirements. So that pressure is an element as well that might complicate transparency. Other factors that came up as relevant to alternative proteins in relation to food authenticity are shown here. Clean label. This is where consumers prefer to see lists of ingredients that are short and the names of ingredients are familiar, some natural, and these might conflict with some of the formulations, at least the ones that are initially used for some products where there is a need to add functional ingredients or to fortify the products with micronutrients. And again, the hybrid composition might mean long list of ingredients. The labeling of the production method is another complication. Some of the methods are very novel, they're unfamiliar, and they are complex. If we think about cultivated products like cultivated meat, lots of things to think about, and the cell types, how have the cell lines been produced, are they using scaffolds, are they edible? So all of these things need to be taken into consideration, and it needs to be well established how to label these things to avoid mislabeling. Intellectual property as well may, in some cases, conflict with transparency, and that might impact labeling. And also the fact that because these products are very novel, and this includes ingredients, supply chains, and this may mean that there is insufficient guidelines, and there are no standards, and again, this can make full transparency piece a bit more complicated. So all of these means that across the different types of alternative proteins, fraud may occur in the same way that with traditional food, we see adulteration, substitution, undeclared ingredients, etc. All of these can happen across all categories of alternative proteins. And here are some of the specific examples that came up during the project. One of them is species substitution, again, where the novel source of protein might be in short supply, they may be more expensive. It's easily envisaged that this type of substitution might happen. Substitution for the traditional source of protein instead of the alternative one. Adulteration with nitrogen powders, that came up as well. Again, powders are easy to adulterate. And then there's the use of unauthorized cell lines in the case of cultivated meat, or microbial species for biomass, or the use of unauthorized insect species. And also the feedstocks that are used to grow these new products. So that could be the ingredients in the culture medium, or the species of microorganisms, or the substrates that are used to grow the insects. And there are other issues like the method of production, for example, the use of wild insects that may not be authorized versus farmed insects. So these are some of the potential types of frauds that the project highlighted. So we can then start thinking what the analytical challenges would be for these type of products. In terms of species of origin, the main barrier is the lack of genomic information for some of them. So new species that haven't been used as food before may not have been sequenced, or the genomic information may not be available. This, of course, be needed for DNA-based methods, or for proteomics methodology as well, where you need this for protein identification. Then the complexity, the complex landscape of novel ingredients, novel production methods, complex formulations. All of these means that even where existing methods might be applied, we are faced with a variety of new matrices that haven't been tested. So this needs to be done before we can understand whether existing methods are suitable or not. And then some of the processing techniques might have an impact on the structure of proteins. So, for example, proteins or other molecules, but if we're thinking proteins, say, for example, fermentation, precision fermentation produced milk proteins, we may think that because initially the protein in principle is the same sequence, we might use an ELISA kit, for example, but we don't know at this stage if the processing techniques are affecting the structure of the protein, whether that particular antibody is going to be able to recognize this target protein, and so on. So yes, discriminating alternative proteins from the conventional counterpart is one of the issues. I've mentioned precision fermentation, but it could be the same for cultivated meat, cultivated seafood, where in principle the genome of the cells cultivated would be the same, but we don't know if post-translational modifications or other features will be different in these products. I've mentioned this as well before, distinguishing wild versus farmed, for example, in insects or in algae. So yeah, existing methods, some of them will be applicable, but they need to be tested and validated in new matrices. And also there's a need to find biomarkers, to create databases, create reference materials, validated methods, all the things that we've been hearing about today, all of that needs to be better defined for alternative proteins. When we looked in the literature, we realized that there wasn't much available at all, and more recently, some more papers are coming up. So I'm showing here some examples that we found for different classes of alternative proteins. In this case, this is plant-based. Spectroscopy and chemometrics has been used to identify powders. So this example here by Neves et al., they were able to classify soy, whey, and wheat as adulterants in plant-based protein powders. There's a company that have developed metabolic fingerprints of commercial plant-based meat analogs, and they've done this by using sensors in combination with artificial intelligence. There is a study by Fowler et al. Where they used PCR, next generation sequencing, and LC-MS proteomics to identify ingredients in commercially available plant-based products in powder form. And this study is interesting because they described successes with all the methodologies, so that there was a certain level of success, but they also found challenges. And this is good to inform how these methods need to develop to accommodate these additional novel products. So one of the challenges that they found was lack of DNA integrity in some of these products. So that was hampering PCR methods, problems with solubility of protein in isolates, making it more challenging to apply proteomics methods. And they concluded that the use of orthogonal methods where different technologies are used to analyze the same sample might be the way forward, and we can use data fusion to analyze all of these different data sets in conjunction to get more robust results. Some examples for insect-based products include spectroscopy again and chemometrics for powders. This was used to identify cricket powder in plant flowers in one of the studies, and another study looked at distinguishing authorized species of insects from unauthorized insect species. They were successful in applying near-infrared microscopy in that case. And there are some studies as well using DNA-based methods. PCR tests have been published for the main species used for food and feed. And more recently, a paper was published where they developed a metabarcoding method. The authors tested, so they developed the method with they claim is a universal primer set that they use for this study, and then they tested it with model foods, cookies and burgers containing insect meal, and they also tested it on commercial insect-based foods. Successful identification in all cases, and soon after that, another paper was published where they used the same primer set to do a survey of insect-based products sold in the EU. And they focused on e-commerce products, so all products bought online, and they found that 1/3 of these products were mislabeled. They were either unauthorized species in the product, or they were cases of substitution for lower value species. So that is a good example of a method that is, the authors have proved that that is working for this type of survey and can be used for monitoring products. And also it gives us an idea of the level of fraud that we might be finding in some cases with alternative proteins. Proteomics has also been tried for insect-based products. In one case, they use spectral comparison to identify species. In another case, they were using non-targeted proteomics, and they found that the success was limited, and again, due to the lack of genomic data for some of the species that they were looking at. And a study by, sorry, my connection went. Can you still hear me? Can anybody confirm? Yeah, okay, thank you, sorry. So POMISAL used non-targeted metabolomic screening. They were actually using this methodology throughout the whole production process to look at quality and certain aspects of their own production system. But they highlighted the fact that non-targeted metabolomic is a very good tool because it can complement proteomics and genomics, and it also looks at a range of different molecules that allows you to identify unexpected molecules, basically. So you're not looking for anything in particular, and that can give you the opportunity to identify things that might be related to safety or other aspects such as wild versus farm discrimination, etc. But again, they stress the need for reliable databases, reliable annotations, and they need to develop in silico tools that will allow identification of unknown features in the data, and also the need for shared repositories. So these are not for studies. They involve a lot of data, a lot of analysis, and sharing is something that is always flagged in this context. Edible seaweed as well is an area where different technologies have been applied. Single nucleotide polymorphisms to distinguish geographical origin, for example, NIR, and chemometrics, again, to distinguish cultivar, geographical origin, or farm versus wild. And elemental profiling as well has been used for geographical origin and for identification of powdered seaweed products. So overall, through the literature review and the conversations that we had with stakeholders, non-targeted approaches came high up on the list because they're powerful, generic approaches that enable the identification of unknowns, and with such a wide variety of products and species, ingredients, etc., they are always considered powerful technologies. Each one of them will have different advantages, but they complement each other, and data fusion, as I mentioned earlier, where joint chemometric analysis can be done using diverse data sets, is something that is considered to be worth exploring for alternative proteins. So the project concluded with some priorities for research and some recommendations that I've summarized here. So one of them is around existing analytical methods. It's worth reviewing what we have and testing alternative proteins, so testing the methods in these new matrices, identify where the gaps are and what needs to be developed further, and then getting accreditation, validation of methods and accreditation with these new matrices. There is a need to investigate biomarkers, and these can be very helpful to answer specific questions, such as can we distinguish a precision fermentation protein from the conventional counterpart? And non-targeted approaches I've already mentioned, and there's a need for databases across all different technologies, and collaboration and data sharing are essential in this field, and also validation validation of non-targeted methods, which is not straightforward and is something that could add value to all of these approaches. And then recommendations about other areas that would support the development of analytical tools. One of them is horizon scanning. So just to be aware of new species, new processes, new emerging analytical techniques so that we can prioritize and focus efforts where it is more optimal. Collaboration between academia and industry is important, and be able to publish data that everybody can access. Certification, things like reference materials, proficiency testing, guidelines, standards, all of these, again, has been mentioned a few times today already. Then there's a need to have approaches to identify and address points of vulnerability in the supply chain, and this is needed globally. So there's a need for international collaboration in this area because the food supply chain is increasingly complex, and with alternative proteins in particular, also not so familiar. So it's all very novel. And there's a need to give access to regulators and enforcement authorities to data and technologies and be able to transfer all of this so that they can use it for enforcement. And again, computing, this has been mentioned. This came up as well as, artificial intelligence, data technologies in general as tools that can add power to the technologies that would help us with authenticity and increasing consumer confidence in alternative proteins. So I'm going to leave it there, acknowledging my colleagues who work in a project with us, and thank you all for your attention.
Thank you very much indeed, Rosario. It's fascinating to hear about all of that work that's happening in a very rapidly evolving area, which is alternative proteins. So thank you once again. And we should now move on to our next speaker, which is Renata Garbarellini, who joins us from the Rauer Institute, where she's led work on developing metabolomics and proteomics tools to identify the provenance of important foods and beverages in Scotland. So Renata, lovely to meet you, and over to you. The floor is yours.
Yeah, thank you. Let me just put here, presenter. Yep. Yeah, I think it's fine now. Yeah. Right. I'd like to thank the Food Authenticity Network for inviting me to present here some of the work we've been doing here at the Royal in the University of Aberdeen. So I'll show developing omics-based tools to verify the provenance of food and beverages. Yes. So ensuring the authenticity and quality of food products is a challenge in the global food industry. There's still a lack of transparency about the provenance of our food that leads not just to economic losses, but also to health, religious, and ethical safety concerns. And there was an increase in the incidence of food fraud that skyrocketed since 2020. We saw like more than 1,000% increase. And then we think, like, what is food fraud? We already heard today, but it's a term used to describe a wide range of deliberate and intentional actions taken by individuals or companies dealing with food to deceive consumers. And there's a lot of types of food fraud: adulteration, substitution of a good quality product for a cheap one, mislabeling, counterfeiting, but also cross-contamination, which can be unintentional, but if a lot of products are processed in the same facility, it can happen, cross-contamination. And then, as already mentioned here today, the case of the horse meat scandal in 2013 that highlighted the importance of the authenticity and traceability. And then maybe you think, oh, 2013, long time ago, but no. For the last 3 years, we still hear a lot of news about cases. For example, the olive oil fraud in the European Union, 9 in 10 samples of honey in the UK fail authenticity tests. Some counterfeit vodka with some compounds that are very dangerous, and also rotten meat mixed with fresh ones sold in the supermarkets. But the thing is, what is happening, right? Why this case increased so much? There are some points that we can think about. For example, since the COVID-19 pandemic, there's a lot of pressure for the economic profit to increase. So some of these companies or people, they try to improve the profit doing some fraudulent actions. Some places there's a weak regulatory enforcement. It's not all the places that have a food standards agents that regulate everything. And also the digital marketplaces, a lot of products that are like sold on internet, is difficult to, the traceability is difficult to see the origin of these products. The climate change, for example, if you import some products for a country and there was some problems with rain, they lose the crops, and then the products you need, you won't have. So to increase this profit, sometimes they mix different things. Organized criminal networks, that sometimes some criminal networks are inside of the labs. So they already know the new methodologies they're doing to test the authenticity. And so they can work around this not to fail the tests. And also what we are talking today, insufficient adoption of advanced detection technologies. So omics-based tools, why they are so important in food authenticity? We talk about genomics, the DNA-based techniques, proteomics, the proteins, the metabolomics, and lipidomics, which is small molecules and lipids. Basically because they have an ultra-sensitivity detection of molecules in the composition of food. There's the ability to analyze highly processed food products, the authentication of food provenance, species, and processing methods, and also some identification of allergens, contaminants, and toxic compounds. As you already heard, like the DNA-based techniques are highly sensitive. The traditional PCR is the standard for species authentication. The DNA barcoding is the most used technique now to species authentication. So they just built this method targeting some genes, for example, the mitochondrial CoI gene in terms of meat species. So then they developed this database, and then they can use these barcodes to see the identifications of species. But there are some limitations of the DNA techniques because of the DNA degradation in processed food when you cook the product. So other techniques came to try to improve this. For an example, proteomics. Proteomics combined with liquid chromatograph mass spectrometry allows for a quick, reliable protein and peptide characterization. It's effective in detecting adulteration in processed and cooked products because the proteins are more heat stable compared to the DNA. It can identify also some species-specific post-translational modifications and generates more reliable quantitative data. Not just proteomics, but also metabolomics and lipidomics can be used in food authenticity. This set of metabolites and lipids is like metabolome and lipidome. They can be affected by environmental factors, such as the geographical origin, the season, the climate, the species, the breed of the animal, and also some food processing. For an example, blending, heating, freezing, thawing, drying, fermentation, that can show significant impact the metabolome. So for an example, if there was, let's say you are analyzing some honey samples, if they heat the samples more than it should, probably some metabolites will show this, will appear. And when you are analyzing halal meat, for an example, there are some metabolites that can be identified that are related to this slaughter process in the slaughterhouses. So you know how this process happen to see if it's really halal or not. So just a few examples. So how it's possible to do this, like to use this omics basis, using analytical techniques such as liquid chromatograph mass spectrometry, but we also heard here today about nuclear magnetic resonance, the gas chromatograph, and also the isotope ratio mass spectrometry. So this is one of the analytical techniques that can be used together with these omic-based tools. And the methods, when we think about the methods, we have basically the target and the untarget method. Of course, there are also some target, but the target and untarget, the difference is when you want to build a method that's target, you require like the precise peptides or the metabolites or the lipids transition selection. So you need to know what type of peptide, for an example, you want to measure. So you need all the mass, the retention time, everything to build this method. It can be a little bit time consuming and needs some skills to do this. On the other hand, untarget methods, it's a little bit easier to implement, but it's much more complex, the identification later. It's a huge number of data, right? So basically, this is the difference between the methods. So in general, you start with an target method, you select some molecules that you think is important, and then you go to a target method. You build this target method to check these metabolites later. Some applications, probably you already heard today, all of them. We are developing here now at the Roet also some methods with honey. We are trying to make some new methods to analyze honey using liquid chromatograph mass spectrometry, probably based just on polyphenol compounds. So you can see the origin of this honey samples based on the polyphenols. Also, we are going to develop some methods with whiskey, but the beverages can also be used in metabolomics. For an example, you can detect some types of metabolites that are present in a single malt Scotch whisk, for an example, compared to a blend one. Also herbs and spices, and meat, where you can use metabolomics, proteomics, and also lipidomics to analyze. Because when you think about meat, each type of meat has its own unique set of peptides. And the exact sequence of these amino acids and peptides is determined by the species of the animal and can vary between different breeds of the same species, for an example. And also different peptides within species in cuts of meat. So you can analyze one if the pie says it's made of chicken breast, it should be just peptides related to chicken breast and not peptides related to thighs or any other meat cut, because then you don't want to buy a product with a premium cut and it's a cheap one. But not just peptides, also different animal species have different types of fat acids in their tissues. Not just fat acids, but also a whole lipid profile, triglycerides, glycerophospholipids, sphingolipids, and also lipidomics can be used to identify the geographic origin. We heard about the isotopes that can be measured to detect the feed or grass feed or cereals feed, but you can do also this from lipidomics. If you have the chains more related to plants, you can see if this animal was fed with grass or cereals. And microbial safety. So different things that you can do with different omics-based tools. And now I'm going to show you some of the work we've been doing with meat here at the Roet. So this is a paper that it's under review. It calls Meat iCode, a comprehensive proteomics LCMS method for simultaneous species identification in meat authentication. So basically, we wanted to develop a convenient LCMS method that exhibits like a high sensitivity and specificity and can simultaneously identify and verify the provenance and authenticity of different meat species in processed meat products. So we developed a workflow to prepare our samples, because one of the bottlenecks in a proteomics is the sample preparation. So we bought some meat from retailers. We weighed this meat around 2 grams. We wrapped in aluminum foil. We cook the meat. We transferred this for tubes, and then we did some washing steps, homogenized the sample, centrifuge, did all the steps, protein assay to see how much protein we had in each sample to digest. So we wanted around 10 micrograms of protein. Then we did some in solution trypsin digestion. We clean up the samples and then we inject in our LCMS. Here is a multivariate statistics. This is a PCA score plot. You already been introduced to PCA. So H dot is one sample. We had like beef, chicken, horse, lamb, pork, turkey, and venison. And we also included 2 samples of raw meat together with the cooked ones to see if we have a difference between cooked and uncooked meat. And here we saw a difference. So we decided that keeping the cooking process during our workflow will benefit, because if later we want to analyze some products, the processed products, in general, they are already cooked or you need to cook. So to see the authenticity later, it will be better just to pick the peptides that are heat stable. So knowing this, we decided to increase a little bit the number of samples. So we went to 8 samples each species for 4 species: beef, pork, chicken, and lamb. You can see here that our method is the reproducibility of our method, because the samples clustered together and separated from each other in terms of species, which means our method worked to separate different species. We did some optimization during our protocol. So the digestion is the most important step when you are working with bottom-up proteomics, which is the one that you analyze based on peptides and not proteins. So in general, the most used one is the trypsin, but some papers show that trypsin is, if you combine lysine with trypsin, you will have a better performance in your digestion. And we decided to test, but here, if you see the results we have, only in beef samples, we saw an improvement in terms of the peptides identified, including the lysine combined with trypsin. But for pork, lamb, and chicken, it says trypsin had a better performance. So we decided that given the high cost of lysine, in an additional step of 4 hours in this digestion protocol, we decided just to keep the trypsin exclusively, because the idea was to develop a method that was like affordable and easy to implement, so a lot of labs could use the same methods to for the food authenticity. Then we select at least 10 peptides each species in our data-dependent acquisition, which is our untarget method. For each species, so beef, pork, the chicken, and lamb. So we had some rules. For example, they should be present in all samples of the same species. They should have an ion score higher than 20, which is how good the peptides match with the database to identify them. They should have 0 missed cleavages and no methionine, and confirmed by the BLAST database. So they should be specific for this species only. What we did next is, so we pick all these specific peptides and we concatenated them, we combined them, making, let's say, an artificial protein. So in a real world, this protein wouldn't exist, but this is our specific protein for beef. So this is all the specific peptides for beef in one protein. And this is what we call our I codes. So each species, we build one I code. So this is our database of I codes. So then our method can see like it's like target method, but then later we use a target approach to process this data. So we can say it's an target method. And then we can integrate this database we created with our meat I-codes in a software integration. For example, the Protein Discovered, which is a software that is very much used. So then the idea is, after integrating this, we, for example, tested using some steak pie, which should be beef only. We got the filling, we prepared the sample, we did the injection, the LCMS, and then we just see if the detection only peptide select for beef, it means the product was not fraud. But if the peptides from any other species appear in our results, it means there's some type of fraud. We also built our target method to compare with our untarget method. So we just select 4 peptides from that list. And then we built this target method. So we got, for example, the precursor ion, the fragments, and the retention times. And we decided to test the limit of detection for the untarget and for the target method to compare. So we spiked some pork in 0.1, 0.5, 1, 2, and 5% in our chicken samples. And then here you can see that we have a good performance of our method. For example, the correlation between the increase in the percentage of pork in a mix, it like goes with the protein abundance. So we could detect an abundance of protein in all this percentage of pork, but we couldn't see a good ion score that was less than 20, as I mentioned before, in a 0.1. So we stated that maybe our untarget method, the limit of detection is 0.5, which is pretty good for an untarget method. And then we checked our target method. For target method, as you can see here, we detect in all of them the 0.1, 0.5, 1, 2, 5%. And you can see the identification in all this. So our target method, we have a limit of detection of 0.1. So knowing this, we decided to test our method, or the meat I-code, in real life. So we bought some products from the supermarket, a range of different products like steak pies, sausage, burgers, kofta, kebab, different things. And then we have the species that were declared in the label. And then we decided to test to see what our method will show, right? And here we found one fraud. So this product, it was saying a mix of chicken, lamb, and beef. We could detect the beef, it was just 1%, but we couldn't detect any lamb. So probably there was a fraud here. And a second product we found, it was this one. The label was saying 6% lamb and 20% chicken. We found lamb and chicken, however, the intensity of chicken was much higher than lamb. So probably the label was not correct, and there was much more chicken compared to lamb in this sample. We decided also to test our method in the target method for some of these products. So we select some products that included pork in their composition and others no. And as you can see here, we could see the intensity in all the products. So yeah, again, it worked for both target and untarget products.
One more minute, Renata. Thank you.
Okay. And then we can see here some applications, further applications of our method. So anything that has a protein source, it can be applied this method. We can have identification of specific peptides is crucial. So this is the most important part. You select the specific peptides. You build these I-codes to integrate with proteomic softwares. It's a high probability of fraud detection without the need of specific primers, like in DNA analysis test. So just in summary, the Omics basic tools are excellent for food fraud detection, as you are hearing today. So our method, it's possible to identify multiple meat species simultaneously in a single run. It detects adulteration levels as low as 0.5%. It's effective for processed and cooked meats. There's the potential to detect food allergens and contaminants, for an example, if you expand this method. And in future research, it's possibility to expand this peptide database to improve the method for broader further authentication, but also combine with other omics tools. For example, we also have data for lipidomics, so you can combine, for example, the lipid profile with the peptides, and then you have like a double layer of protection to see the authenticity of this product. I just want to say thank you to the RISAS, the Scottish government, for the funding this work we've been doing here, the University of Aberdeen, and of course, all of you. And feel free to send me questions and I will be happy to answer later. Thank you.
Thank you very much indeed, Renata, for taking us through that journey on omics. It's absolutely fascinating. Thoroughly enjoyed your presentation, and hopefully you maybe ask some more questions shortly. So do stay. Okay, we're now through to our final presentation of today, and it's my pleasure to introduce Arne Dubecker, who's joining us from Germany. Arne is the head of the Titamus Center for Food Fraud, and he's going to be talking to us about untargeted methods to identify markers. Welcome to the conference. Over to you, Arne.
Thank you. So I hope by now you should see my slides, right? I'm sharing them. No?
Yes.
Yeah, you do see them. Perfect.
I do, yeah.
That's
perfect.
it. So yeah, first of all, thank you for having me here today. It's really a pleasure. Very interesting talk so far. I really enjoyed it. And I will talk about also non-targeted analysis for a couple of applications. First of all, maybe a small short introduction of myself. So my name is Arne Dübecke, and I'm working for the TCF Squared. That's the Tentamus Center for Food Fraud. And the juice is one of the centers of expertise of the FAN, and yeah, one reason why I'm here today. So we are basically a bunch of private labs working on food analysis, and authenticity is my topic basically. So it's one of the things we do. I have about 4,000 colleagues worldwide in 22 countries, and altogether, we are about, including other labs like pharmaceutical labs and so on, we are 97 locations at this point in time. So yeah, if you look at this thing here, you might think maybe this is a laser gun of Star Wars or something like that. Like the Lego version here, it does look kind of similar, but nope, it's actually one of the first LCMS that was equipped with electrospray ionization. And this electrospray ionization is still nowadays very widely used in many applications. Also at Tentamus, we use it a lot, of course. You see there you have simple peaks. It's very easy to separate the peaks there and to grab the information from the chromatogram there. Since then, of course, a lot of things have changed. It got much more complex. We added dimensions to the analysis from GC to GC, GC, LC to LC, LC. We don't just look at the parent ions in mass spec, we also look at the fragments, and sometimes even at the fragments of fragments, so you get more and more information. Ion mobility has been introduced a couple of years back, and we switched from unit resolution for mass spec into high resolution, which comes with a lot more data than we had before. So all in all, it's not really possible with these high amounts of data to just check with your eye and look at some of the peaks and derive your conclusions from that. So we are now looking into patterns and fingerprinting mostly. Which of these approaches, targeted or non-targeted, you use? Of course, it depends on what you want to do. If you know what you're looking at. If it's a certain compound, you know the name, you may be even able to buy a reference material. Targeted is still the way to go. You look very specifically on one compound, while of course with a non-targeted and all this data it generates, you are able to have this 360-degree view on, not on everything in your sample, but on quite a lot of different things in your sample. Of course, these things come with pros and cons. The targeted is relatively simple, or it's actually compared pretty simple. There's no statistics involved. You just have the peak, you can integrate it, you can calculate quantification, or you can quantify a substance. You can, and that's actually a pretty good advantage of targeted analysis, really nicely fine-tune each compound for maximum sensitivity. You have a reference standard usually, you can tune it in and find collision energies and all these kind of things very specifically for your compound, and gain the highest sensitivity possible for that. You generate low amounts of data only because it's just it's not too many things analyzed actually, so it doesn't require so many data. You can get, or if you can get a reference standard, which is possible for many substances, you can easily quantify it, and you don't need even a database for that. Opposed to that, if you look at the non-targeted, it's pretty complex. You need sophisticated statistics to evaluate this vast amount of data, because as I mentioned, just by looking at it with your eyes, you won't see anything. It's too much. You need these statistics for that. And a little downside of that is, as you analyze all these substances in one run in the LCMS, you cannot really optimize for certain compounds. So you are taking like an average method, which should work for most substances okay, but not necessarily great. And this can lead, of course, to some substances not being very sensitive or detected very sensitive, or not at all, maybe. You generate a lot of data. When we bought our high-resolution mass spec and started analyzing, one run usually ended up in 500 megabyte of new data, which is quite a lot if you consider that you just don't measure one sample per day, but a couple of more usually. So it's a lot of data being produced there. Quantification of the unknowns, if you don't know what you're looking at, then of course quantification is not really possible. And in order to derive conclusions from what you're analyzing there, quite often you need then the database. The downside of the targeted analysis, of course, is you look straight forward, you don't look left, you don't look right, and you can't look backward in time, for example. If you're interested in figuring out something about another compound, it's not possible, especially when it has a different mass to charge, then it's not possible to look into old data to check if it was there already. With the non-targeted fingerprint instead, you can detect unknowns. So if you find a peak in there that might be of interest and you find it today, but you measured already lots of other samples with the same setup in the past, you don't need to reanalyze anything. You just look into the old data because all the information was in there already. It's just that at that time you didn't know about that compound, so you didn't check for it. But this retrospective data evaluation is possible in this case with a non-targeted analysis. It does come with challenges, especially when you think of standardization. Like as you usually need databases, a database has to be created with the same kind of analysis in the end. So the sample preparation has to be standardized before you start building your reference database. So this is some work that you need to put in there to figure out the best kind of sample prep where you get most substances that could answer maybe your potential question are contained in your sample and your sample prep. Same thing also for the actual analysis, the instrumental analysis, the mass spec settings or HPLC settings. You can't just, if after a couple of hundred samples you think, oh, I should maybe take another chromatographic column, you can't do that. Otherwise, you would have to start all over again with the analysis. So all these things you need to think of before actually starting. Once everything is set, then harmonization of interpretation might be also hard because as there's a lot of statistics involved, there's not really, or quite often not, a sharp line between a yes and a no. So there's always a certain room for interpretation. And especially when you think of commercial labs, different companies doing more or less the same, but sometimes, especially in borderline cases, you will have contradictory results. So this is not easy. That's actually a bit challenging sometimes. Yeah, the quantification of adulteration in general is not really possible unless you can see maybe a dilution effect or something like that, because usually, even if you find a marker or something that tells you, okay, this is adulterated now, you still cannot tell how much adulteration took actually place, usually, because for that you would need the material that was used for adulteration, and usually nobody's giving that to you. Yeah, and reference data, or samples, reference samples. We heard that before. This is a big issue. Of course, you need good or authentic, especially the authentic samples. Getting them is not so easy, but yeah, well, you need some of them at least. But in the end, yeah, it depends on the question you ask, what you need, if you need targeted or non-targeted. For example, if you know that adulteration occurs, you know the type of fraud in the affected matrix, and even the adulterant, then you just can go for target analysis. For example, if you analyze Sudan dyes and palm oil, you just have a couple of Sudan dyes, you get the references, you can go straight ahead. If you want to know what is left and right, if there's anything else in there you didn't think of, then of course it gets more complicated and you might need to start the non-targeted and analysis, and yeah, finding out if there's anything else maybe in that palm oil that is used for adulteration. And if you have if you're starting a very new topic, maybe a new matrix, so you don't even know if the matrix is usually a victim of adulteration, and you don't know how it's adulterated, then you really need to start with databases so that you know how actually a good sample looks like, to be able to find out if there's something going on in other samples that shouldn't go on. If we look at fruit juices, actually, there's an organization, the AIJN, the European Fruit Juice Association, and they published 29 reference guidelines for 29 different fruit juices. It's all targeted parameters where they give ranges, not just the amino acids that I picked out here. There are lots of other parameters, and also very different ones, like from trace metals to the amino acids, sugars, and other substances. And though this is all targeted, it is already quite a strong specification, because it's really quite a number of different values that you can check or compare with to derive the authenticity of a sample. And there's one nice tool, it's called juice profiling, that was developed by Bruker together with SGF, and SGF is an industry association with members from industry who control the juices for authenticity and other things. And they use the NMR, and it's a combination here of the targeted. You see here the little table in the middle. Some of these parameters are targeted and quantified, but they also combine it with non-targeted analysis of the entire spectrum to see if there's anything wrong in the sample, or you get classifications also of origin, either botanical origin or maybe geographical origin. The same thing, this profiling, there's one for juice, there's also one for honey, and also for wine and olive oil. Here, two pictures from juice and honey. So what I like about these is this visualization using the quantile plots. Here you see the colored band is representing the database data, and the bold black line is representing the actual sample in question right now. And as long as the black bold line is close to this red band, everything should be fine. It looks like really well in the database data. But here in both examples, for fruit juice and for honey, you can see both samples are very far out. And I like that so much, because it's this, with all this data involved, this gives you a nice visual feedback, easily understandable. You see it's out of the colored band, it's fine. So even with all statistics behind, it gives you a nice picture. So that's why I personally like it a lot. Yeah, with all of these things, you need reference samples, as I mentioned before, which is the hardest bit because apart from the fact that they should be authentic, especially for the authentic reference, it has to be authentic. You need to check for natural variabilities. For example, if you look at orange juice or different orange varieties, how different are they, or geographical differences. If you have an apple juice from Germany and apple juice from Poland, do they look the same or not? So these things you need to learn. Seasonality, you have one year very dry and the next year it's very wet. Does it change the profile or not? Or how significant is this change? So all these things you need to check. And if you're taking samples from suppliers, you need also to check the supplier or keep in mind that you have a certain supplier base and a certain bias that comes with it. So if you have a different supplier base, maybe they come with completely different samples and things look different. So these are all things, or some of the things, there are more, but some of the things you need to keep in mind. At KLab, that is the center of expertise for the fruit juice analysis here in Germany, in Hanover. We did a project some years back on mango authenticity. We used NMR in this case for that. The aim of the project was to identify if to mango puree from Alfonso cultivar, if there's any other cultivar of mango, but all mango added. Alfonso is the most expensive one. That's why it is of interest to find that out if there's a cheaper variety mixed in. And it was actually, so it was a non-targeted approach. And from there, using PCA, we could actually arrive at a couple of markers, like the niacin, trigonelline, and histidine. Elevated concentrations point to or can be found in the Alfonso, while alanine levels are usually pretty low in Alfonso. But also using the non-targeted fingerprint the complete fingerprint was using that, it was possible to find admixtures of the other cultivars to the Alfonso puree. Yeah, here on the left side, that was actually in 2018 when we bought our first high-resolution mass spec, and this was just a trial I did to see how well everything works. I basically just grabbed a couple of samples from our honey storage room, so some Manukas and then some whatever, any other kind of polyfloral or other kind of honeys, just to see how it groups. So it was really a standardized method, dilute and shoot, quick and dirty, and just analyze quickly within, I don't know, over the weekend or something like that. And on the Monday, I did a quick PCA with the software that came with it. And you can see that it was already separating the Manuka very well from the rest. So that was just the first trial to see if actually a high resolution mass spec is something useful. And well, we figured it is useful. However, we just had at that time one high resolution mass spec and several triple quads in routine analysis. So the idea then was basically that we gathered a lot of honey samples, authentic ones, and also a lot of the syrups that are usually used to adulterate honey. And then we analyzed it all with a high resolution mass spec and a mass from, I think, from 100 M/Z to 1200. And then we just statistically compared to identify which mass to charge values or numbers we find in the syrup that we do not find in authentic honey. And that is then basically our marker without even knowing which substance is behind. It's not necessary as long as we know that it doesn't occur in natural honey. So we came up with a couple of markers and the MRMs, so the mass to charge ratios, and the fragments, we just transferred it to our triple quads. And yeah, with that, we could then analyze quickly and relatively simple in routine for the authenticity using these markers derived by high resolution mass spec. Another reason for that was also that the approach HRMS and normal triple quad LCMS targeted is very different. So you can't usually just take someone who's done for years targeted analysis and just put him on a high resolution mass spec to do non-targeted. It's not that easy. It will take a while for the mindset to switch. So this is maybe also something to keep in mind when you start doing stuff like that. Yeah, for the same reason that the high resolution mass spec is not as much available as the normal triple quads, we just thought, okay, for fruit juice, most likely the same syrups would be used for adulteration. So the syrup markers that we actually found for honey should work, or in principle should work for fruit juice as well. And also resins that are used in some countries for fruit juice processing and also for honey processing should also be the same markers in there. So we should see some overlap there. So the idea was just then basically to copy paste these data to the juice environment. Of course, with some drawbacks, which you wouldn't have if you would just start straight away non-targeted with a high resolution mass spec. And it comes, of course, also with additional work because you need to verify at first, are there any markers? Or no, you have to verify that the markers you took from honey, that they do not naturally occur in fruit juice. Because this would be, of course, not good if you find everything's adulterated, although it's a natural substance. So this is something you would have to make clear before you start. Matrix effects will most likely be different from honey as well, and a substance or a marker substance that you will nicely see in a honey matrix might not be visible in the fruit juice matrix. So this is another point keeping in mind. Of course, also maybe some of the honey markers are not of interest for fruit at all. Could also happen for several reasons. And another thing is if you cannot add with that, of course, new markers to that. So you have to stick with the marker set you have. Maybe you have to reduce it a little. If you find that naturally these compounds do occur in fruit juice, then you would have to reduce it a little bit. But you cannot just add, you just have to use what remained there. And if you really want to extend the marker set for fruit juice, then you would actually have to start the high resolution mass spec database again. We are looking at a broad mass to charge ratios range in quite a number of fruit juices, and then you can derive new markers, of course. So one thing that we finished, a project that we finished recently, that was actually a funded project funded by a third party in Germany, collaboration together with the University of Stuttgart with Jens Brockmeyer. We looked at, we tried a proteomics approach to analyze honey, also for authenticity. The idea is basically that when honey is produced, then the bee is giving a peptide or proteins into honey, and if we figure out which are the key peptides that we have to look at, we can quantify them, we can establish normal concentration ranges of them, and then via dilution effect we could then detect adulteration independent on the syrup. Because this is, of course, if you look at the marker approach, this is a downside. A new syrup which doesn't contain markers, the markers you look at, you will not see it. And this is what actually usually happens. That's also the reason why nowadays more and more these statistical methods are getting more important, and they are more accepted actually. One reason why we didn't use that 6-7 years back was basically that the acceptance among clients was not very high. If they used to like answers like, yes, my sample is within specifications, or no, it's out of specifications. If you told them, okay, with a probability of so and so many percent, it is maybe okay or maybe not, this is not what they want to hear. So we try to reduce the amount of statistics displayed to clients because it wasn't very well accepted. But this is, or seems to change, I think, because everybody now is realizing that this is most likely the way to go. We're getting even more data than before. Now we have, as we've heard before in the talks, deep learning, we have neural networks and artificial intelligence, and all these things are helping, and they're becoming more and more popular. So yeah, this will be the way to go, going more into statistics also, and not just the markers. But the markers are good for simplification, for not having to buy too many of the expensive machines, because a high-res mass spec is about twice as expensive compared to a triple quad, which is of course quite something. Now back again to the proteomics. So it's basically, yeah, we took the protein out, we did a tryptic digestion, and then a non-targeted analysis of the proteome or of the peptides in there, and that was done at the University of Stuttgart. And 44 peptides were identified, and yeah, so it is possible to see a certain of dilution, and also other things like, for example, the botanical origin is also apparently for three types, at least for 3 types of honey we tried it, it's possible. So that's more a proof of concept. It seems to work, and this might be very interesting to have a deeper look into that in the future. Yeah, so with this, I am coming to my end. I thank you very much for listening to me, and I hope you enjoyed it as well. Thank you.
Thank you to you, Arno. I did enjoy it very much. So thank you very much for presenting to us. That was really absolutely fascinating. So we're now moving to sort of the last phase of the day. First of all, I think I'd like to take this opportunity to thank all of our speakers today for their fabulous and insightful presentations, and really sharing what they're doing to advance food authenticity analysis to help us make sure that consumers can have confidence in our food, we can have a resilient supply chain, and that also that we can keep ahead of the fraudsters. So that was great to hear what you had to say today. So we're now going to move into the Q&A panel session, and I'd like all of today's speakers to please switch their cameras on and unmute themselves, and then I will attempt to try and put some questions to you which have been very kindly collected from the team who are supporting the event today. I'd also like to take this opportunity to thank the audience too for submitting your questions to the panel throughout the afternoon, and we've tried to group them and cover as many as we can. So I hope that is okay. So the first question I've got is actually for you, Rosario, which is this was from John Robinson, and it is, for novel protein sources, particularly insects, have you looked at the risk of allergenic reactions for the consumer?
Yes, I've seen that in the chat, and that we've kind of different people have added bits and pieces, but yeah, I can summarize. So that this is certainly a concern, and we've not looked at allergic reaction specifically, but we've looked at other aspects of the potential of insect proteins in terms of allergy. Helen did a project recently looking at whether current methodologies to detect allergens would work on insect proteins. So if you use tools that have been developed for crustaceans, could they work for insect proteins? Because this is known, so the cross-reactivity between insects and crustacean is known. So I think the only way of knowing for certain would be to do human trials, but we also published a paper looking at homology between insect sequences and crustacean sequences, and there are many other papers in the literature. So this is something that is known. And what I do know, and again, this came up in the conversations that we had with stakeholders for the novel for the alternative protein project, is that insect food producers do label the products as may cross-react with crustaceans. So they have a warning for consumers who might be allergic to crustaceans. So yeah, at least for insects, there is this cross-reactivity issue that is well known. But with other alternative proteins, it might not be known. With something that is completely novel, there isn't. But the novel foods regulations demand that an allergenicity assessment needs to be submitted with the novel food dossier. So that at least doesn't give you 100% response, but at least it gives you an idea of whether a certain novel protein might be a risk in terms of allergy.
Okay, thank you, Rosario, for a very comprehensive answer, and I hope that was of interest to the person who put the question. So Helen, I've got a question for you. This is from Catherine Cockcroft, and she wants to know, is it possible to determine a particular plant cultivar, e.g. Alfonso mango, which carries a premium price over non-Alfonso mango?
Yeah, it depends, I guess, on the genomic difference. I know very little about mango genomics, I have to admit. So yeah, it depends, and I don't know what makes it carry a premium price. I don't know if you're the person who's asked the question can speak a little bit more, maybe, and I can get a better context.
Okay. Well, perhaps we'll see if they put something in the chat, and we'll move on to another question. So if I go to you, Kate, some of the aspects that you've spoken about in practice about robust untargeted databases can only be built and accredited by teams. Can they only be built or accredited by teams who have expert data scientists, or can analytical chemists do it alone? So I think the question is, can analytical chemists do what you were talking about alone, or do they need to include experts who deal with data?
I would say that analytical chemists can do it alone if they train themselves up to know something about multivariate statistics. So I suppose I'm saying that you do need to have some statistical knowledge. I think the misuse of statistics, not just multivariate statistics, I mean any statistics, but especially multivariate statistics, is so easy to do that I think using it with absolutely no background knowledge to call upon would be not advisable, because I'm not aware of any user interfaces, any pieces of commercial software which would allow you to do flexible modeling without any risk of doing it badly, because there's always some things that you need to tweak and parameters you need to set. And just blindly setting without knowledge of what they are. So I think you need somebody to train up so that they know the basics. Does that make sense? That would be my view.
It does to me. So thank you very much, Kate, for your view. And so, Renata, if I turn to you now, this is an interesting question. Given what we heard about DNA methods today, how do you think proteomics can proteomics be more useful than DNA methods, in your view? So it's kind of, I think, which one, or what's your view on that? And that was from Zara.
Yeah, I think in proteomics there's the benefits that I mentioned over the degradation of the DNA. The problem is that proteomics is still not fully automated as the DNA-based techniques. So there's still some problems, as I think Rosario Allen mentioned, about the standard methods. So sometimes if you have the same method and other labs just try to reproduce using different instruments, and some of the things, sometimes it's different to compare or having the same results. So I think proteomics can have this potential, especially the target methods. I think it's easier to have this inter-laboratory. But if you just look for the untarget methods, I think there's still there's a little progress that still needs to be made to achieve this. But I really think that, yes, it can have the same confidentiality comparing both methods.
Okay, and it looks like Rosario would like to come in. Do go ahead, Rosaria.
Yes, please. if I can have a moment, because if you remember, and Sophie mentioned this project in her talk this morning or earlier today. So we did a project, a similar approach, very similar to Renata's approach, looking at targeted methods, but for a range of different species. And we cooked our meats following an industry recipe, and we tried really highly processed products such as corned beef. We did find cases where proteomics could identify the right species and PCR couldn't. But in general, of course, PCR, you're right, Renata, is easier perhaps as a method and quicker, especially the sample preparation part. But it's a complete, proteomics is a robust methodology as well, and in some cases it could give you a response where DNA is highly degraded. So I agree with that.
Yeah.
Okay, thank you. That was really helpful. Thank you. So if I turn to our colleagues from government who are on the line now, and if, for Bhavna or Sophie, either of you, what do you think have been the main shifts in the type of authenticity issues that you've looked at over your various programs over the years? So how have they sort of changed over time? Go on, Sophie, you were first.
Happy to go first. I think probably just building on the response to that last question, really, that quite often we now need a sort of multi-method approach of different techniques to look at complex issues. So you might use DNA and proteomics approaches, or metabolomics and proteomics approaches as well. Whereas I think previously we focused quite a lot on, single confirmatory methods, particularly based around DNA. But I think things have moved on and shifted in that direction. I think also, given there's a lot more, it feels like there's a lot more commercial laboratories offering methods to meet sort of market need. A lot of the work we do under the programme now is around providing sort of best practice advice in application of methods, in kind of how they can be standardised and validated, which is a bit of a shift from kind of the work we used to do before. And I think also the move from sort of targeted approaches to non-targeted methods has meant we've done a lot more in that space around databases, database curation, database building, and assessing database fitness for purpose. So a sort of a move to do more on advice, drawing on our expert committees as well.
Okay, thank you. Bhavna, is there anything you wanted to add from your perspective?
Yeah, so I mean, the two reviews that we did on the methods for herbs and spices and the country of origin did say that a combination of methods is useful. I mean, well, one of the things, and I've just seen a question on the chat about how can we make sure that these tests are approved third party. So I mean, what we sort of, what the legislation says that labs need to use accredited methods as well. So as Sophie mentioned, there's a lot of non-targeted screening methods out there. How do we ensure that they are quality assured and, accredited? Because that's the bottom line, that methods need to be accredited. So I suppose when you're using screening methods, you still need that confirmatory testing as well. Yeah, it's a minefield out there, but I think it's good that we are shifting. We're looking at new technologies as well, where we didn't, have these sort of technologies in the past, but there's a lot of work involved, and I think the other thing is where you cannot get that definitive method or the results, then there are other approaches that we consider with our local authorities, is using sort of traceability and weight of evidence as well of the sample. So not just looking at the analytical result, but looking at tracing the product back as well.
Okay, thank you very much for that, Bhavna. That's helpful. If I may ask you another follow-on question, if I may, you mentioned sampling in your presentation, and what do you think are the key challenges that we're going to face in the future in terms of doing sampling and surveillance?
Yeah, I think for us, well, it's funding. So it's, you know,
Yes.
how much sample can you take when you don't have that much funding? So this is the reason that we are sort of targeting our sampling. So we don't go and sample everything and get everything compliant. So we do target sampling at risk foods and commodities. We've, you know, so I think it's just making sure we've got better intel, better horizon scanning, those sort of tools to help us know where to target our sampling. And also I think collaboration as well. So, you know, making sure that we talk to industry, we talk to other government departments, local authorities, just, you know, just to make sure that we consult and we know what we're sort of targeting. But yeah, so I think a better sort of collaboration and making sure that we're not sort of duplicating as well our efforts.
Okay, lovely, thank you. Anna, if I could turn to you, you presented very nicely the sort of pros and cons, if you like, of targeted methods versus non-targeted methods. What's your method of choice?
My method of choice. Well, I did work a lot with NMR and I did work a lot with LCMS. So they are definitely very powerful methods. But in the end, it depends on what your question is or what your matrix is as well. Especially if you look at herbs, for example, and what the other plant materials are in your sample. So that is also a very powerful method, though not exactly non-targeted. It just contains heaps of targeted targets. But I think this is a very good method as well. But yeah, so there are many different ways I think you can use this non-targeted. Also, for example, if you have handheld methods, there are some handheld options where you can check in the field, which basically, like FTIR, for example, they might be not as good as if you have a proper lab method or method set up in the lab, but it's still of use in the field to get a relatively quick answer if something is okay or maybe not okay, or maybe either completely out of spec or at least something where you should have a closer look to it. So I think a real favorite method. So for myself, as I said, LCMS and NMR, or high resolution mass spec and NMR, I really like that because I worked a lot with that and it is very powerful. But there are many other ways you can use non-targeted to get useful information.
Okay, that was really nice. Thank you, Anna. Renate, if I could just move to you, sort of sticking with methods for a minute. How close do you think omics type techniques are suitable for commercial high throughput lab testing? So I suppose, almost like a point of use type test.
I think these are also already mentioned, like, for example, if it will be good to have a sample automation of the process, like, because sometimes the way you process the data can be like a little bit different. And if you have this, and then to process the data, to do these workflows, so nowadays, we already have some like robots that can prepare the sample, that can automate this part of the process, but still the data analysis is a little bit difficult. So I think we are going to this. The DNA-based methods, they are much more high throughput nowadays. Maybe some target methods like developing the untargeted and move it to a target. It's much easier for a laboratory, for an analytical chemistry laboratory to do this instead of the untarget. For this is why in our method we try to do this, use the untarget approach, and then you targeting the process data later to help with this process. But I think we are moving in this direction. For example, metabolomics, you already have some companies, they are doing some kits that you have a list of metabolites that you can target using this kit, so it's not that complicated. I think lipidomics, we still don't have a lot, but I think the omics, they're going to this direction. So it's still, we still need a little bit more standardization in terms of methods and everything to reach this. But I think in a few years, maybe we will have more.
I love your optimism. Okay, thank you. Helen, in a similar vein, just to ask you, you talked about country of origin. How do you see country of origin developing into sort of being an almost like commercialized, easy to use test? Do you see that happening?
I think for emerging technologies, there's a lot of potential in spectroscopy methods, maybe FTNIR, and they're nice handheld, non-destructive methods that can be done rapidly in about 5 minutes. I think that would be a nice screening method for a lot of commodities. And then as suspect samples are thrown up, then other technologies will be implemented. But there's a lot of potential for spectroscopy going forward. Developed on high res probably is the way to go forward, and then taking that down to handheld methods.
Okay, thank you. So I'm going to move sort of away from methods and more towards adoption. And this is for any of our speakers, I should say, to answer. So what steps could be taken to encourage the wider adoption of technologies such as DNA barcoding to strengthen import controls and consumer confidence in the UK market? Maybe no, I don't know if we have any DNA barcoding experts on the call. Sophie, go for it.
No, I'm not a DNA barcoding expert. I just wondered if that might be a better question for tomorrow's session, since we have a talk on NGS and sort of DNA-based techniques.
Okay.
But I think probably the answer is sort of making them quicker, cheaper, simpler, faster to run, but that's not always easy to do, and making sure the sort of databases that maybe underpin DNA methods are robust.
Okay, I think that's a fair challenge, and we shall certainly take that away for tomorrow, because we've got lots of experts we can put that question to. Okay, so another one for any of our speakers. How do you think we can move from a weight of evidence approach, which we've talked about a little bit today, to the sort of legal threshold of beyond reasonable doubt? How do you think we should try and tackle that?
I can start off on that. I think a weight of evidence approach at the minute is working reasonably well for some of the work that I've spoken about. If you present to suppliers the data that you have, and if you've got good reference materials, certified reference materials, and good databases, that data is underpinned by a lot of producers step back at that point, and a judicial process isn't needed usually.
Okay, thank you, Helen. And then, oh, Sophie, would you like to come in?
Yeah, just I suppose to add to that, I agree. There's lots of different, I suppose, steps in the enforcement hierarchy, so it's not always going to a court case. But I think weight of evidence approach isn't going to go away. I think it's necessary in a lot of cases. And think where you don't have a confirmatory test, a single test. And I suppose I don't know that there's necessarily incompatible between strong weight of evidence and where something has to be beyond reasonable doubt. That's probably more for someone who works in the enforcement area to comment on, but I'm not sure those two things are necessarily entirely incompatible.
Okay, thank you, Sophie. And I think this is probably going to be my last question, which is for Barbara and Sophie, I think. So, which is, how are regulators leveraging or using some of the technologies that we've heard about to better protect the consumer? That might be more for you, actually, Barbara.
Yeah. So, I mean, I did mention one of the case studies where we are trying to sort of implement the sort of more screening type of methods into the laboratories. Yes, it's quite tricky when it comes down to funding as well, and the labs need to keep their methods accredited. So that's where all the challenges are. But yes, I mean, we are doing a lot more work on non-targeted methods and seeing how we can add these to the labs. The other thing is that laboratories already have instrumentations that they're using for other tests, so non-authenticity tests. can they be sort of adapted to look at authenticity issues, and vice versa? So, you know, at the moment, you know, the PCR, real-time PCRs are in the labs. Can they be used to look at allergens? You know, so actually it's making the best use of what instrumentation is already in the labs, and you know, to sort of make it more sort of cost effective as well. But it's good to sort of hear about the proteomics as well, because that's something that we need to explore a bit more with the labs.
Thank you very much indeed. And thank you to all of you for answering all those questions so eloquently. It's much appreciated. I think where we are now is I'll probably give you a few reflections, if I may, from today. So I think it seems like a while ago, but it was only this afternoon we heard from Gideon, because we've had so much fantastic information. So we heard from Gideon Henderson, Professor Gideon Henderson, about how DEFRA uses its evidence and science for policy making and addresses risk, and he talked about some of the challenges around the food system from now in the future, and how we use science to address those. And then he talked a little bit about the role of origin, which we've heard and how we could test for that, and also some of the DNA challenges, and some of the challenges rather that we've used DNA to tackle. And then we moved on to hear from Sophie and Bhavna, our government colleagues, who gave us a lovely sort of journey around the methods of the work that both DEFRA and FSA do collaboratively, from method development to rolling work out to laboratories and working collaboratively. And also importantly, the area of harmonisation and standards, which I suspect we might talk a bit more about tomorrow. So it was really insightful to hear about that. And then Kate talked to us about machine learning and AI and reference databases and things, and random forests, which I'd not heard about before, and I found that absolutely fascinating. Again, a new and exciting area that I think is something that we certainly should keep an eye on for the future. And then we heard from our colleagues at FARA. We heard from Helen, who talks about the country of origin and the work that FARA have done on stable isotope ratio analysis, and some of the challenges around that, particularly around the database challenges and the importance of having authentic reference samples. And then Rosario, you talked about alternative proteins, another emerging challenge, I think, and some of the challenges around testing for that. Then Renata, you gave us a nice flavour around sort of drivers, if you like, for food fraud, including economic losses, climate change, and all sorts of things that we know impact on why food fraud happens. And then we had a discussion around some of the genomics methods that you talked about. And then lastly, when they talked about the work on sort of comparing standardized and targeted and non-targeted methods for us, and we had a really interesting look into some of the work that he's been doing, including on honey. So I think, in conclusion, I think we've had a really, really good look in the authenticity toolbox today. It's been fascinating, and I hope you've all found it fascinating too. And I would just like to thank all the speakers again for all their thoughts and insights that they've shared with us today.